Testing. Okay, perfect. Thank you. Awesome. So it is 11 o'clock. Thank you so much, everybody who's returning. I'm seeing a lot of familiar faces. So this is the second session for the AI session. Once again, thank you so much for being here. Speakers will have 10 minutes for talks and 3 minutes for questions, and then we'll have 2 minutes to switch back and forth. If you are a speaker, you will, in person, you'll have a little light system up there, which will remain green for the first 10 minutes, go to yellow for the last 3 minutes, and then once it turns red, we will start moving speakers. So my name is Kit. I will be moderating this session that was organized by myself and Rukeya Johari. And yeah, if you have any questions before we get started, please feel free to ask. But if not, I would like to welcome our first speaker, Kai Kao, and the talk will be on enhancing species retrieval in Flora through language model integration. So, welcome him up to the stage, please.
Thanks, TechWeek, for giving me this opportunity to this present. Hello, my name is Kai, or you can call me Derek. I'm from Taiwan, National Pingtung University Science and Technology. This is my title, Enhance Plant Species Retrieval in Flora through Language Model Integration. And Zhikai Yang is my advisor of Department of Forestry, and Jianxin Chen is a teacher who taught me Python and machine learning. First, I like to talk about two plants retrieval from two perspectives, name retrieval and the trait retrieval. Name retrieval involves querying information based on known species name, which is what most biological information platforms offer. And trait retrieval, on the other hand, involves inferring a species based on its known trait, and that's the focus on my talk today. The most common used method is the dichotomy keys, widely seen in taxonomy literature. It's excellent for distinguish between species, but it's not beginner friendly. The trait listed might not match what the user want to query. And the interactive key produced by the data system is another method, which involves selecting and inputting items provided by taxonomists to filter out species. However, to complete this interactive key, it actually requires extensive manual definition of char, item, and spec files before it can run. This undoubtedly costs lots of time. Yes, these methods are academically precise, but they are also their drawback. They are too rigid. In data science, there's no Boolean retrieval method where the result is either true or false. Since plant traits often vary, a slight deviation in the user's query could include relevant results. they doesn't figure out the problem of semantic similarity. Like the system will never know how long circles across their similarity they are. At this point, language model can come in handy. After extended training on textual data, the model can understand this kind of problem. By calculating the cosine similarity between two sentences based on their vector embeddings, you can determine the angle between them in a vector space, which represent their semantic similarity we need. When generating embeddings, we can choose any open source GPT language model, like OpenAI's GPT, Google's Burst, or Meta's Llama. And I use the experts in this project. Here's an example of contents typically found in taxonomic literature. I'm certainly work with a data set with 2,675 species common to both in Flora, Taiwan, and Flora of China. In practice, I extract this textual data from the Flora, like this, and I pass it into this format. And how does it work? And I think everyone can do it by yourself. It's definitely not done manually. I just tell the ChatGPT, please organize it into a table, and it's done. Yeah. In this step, I didn't use any API keys because I'm the low-cost guy. So I wrote a custom crawler script to complete this step, the Python Selenium. Yeah. So once I have the data from the Flora and the user input, so I convert it into embeddings and calculate the cosine similarity. The previous samples are just the score of all traits of one species. However, I should calculate these traits of all species in the database. The traits with the highest score for each species are highlighted in red. If a user inputs multi-traits, we average the highest score trait for each species. If you don't understand, but more friendly to look at the code in Python, you would first group by and find a maximum value, average the score of each column, and you can get the result. In the conclusion, I want to emphasize that in this case of trait retrieval, were traditionally considered Boolean retrieval as the default system, requiring us to define every trait for every plant organ. This is consumer need persist that's involved instant discussions with taxonomists. And I apologize that although this section are supposed to be talking about standardization, but it seems I'm going to be the upside direction. I abandoned the need to define larger number of the column. Instead, I pass the text data to use language model embedding to complete the similarity calculation. Look ahead, most research certainly focus on morphology and ecology, exploring trait, species, and their environment. However, there's a limited research on functional trait that connects this accept. I believe this is due to lack of a large-scale trace database, at least in Taiwan. Countries like China and Australia have already made progress in this area. I hope to see trace research from a broader perspective in the future. Finally, I want to thank everyone who has helped me along the way. I have put my project on the Hugging Face. You can try it. Thank you.
Awesome. So we do have a moment or two for questions, if there are any online or any in the room.
Hi, Derek. You use the average mean to compare the similarity with Why do you choose the average mean instead of other criteria like geometric mean or others?
And I organize the table. There is no geographic data but I can input in the future. I still not input the geographic data yet.
I say geometric mean, 集合平均 geometric. Geometric.
Because I think cosine similarity is more can know about semantic.
But you didn't know how the vector space, how it looks like. You don't know the distribution, right?
Vector about.
Okay. Thank you very much.
We have a question online. What challenges are there, if any, to scaling AI keys up to tens of thousands of species?
Scale tens of. I write a crawler script to the ChatGPT and get response by myself. So I just like a robot and see the scan and watch and check the ChatGPT response is not getting wrong. Or if it's getting wrong, I stop it and restart again. And there's no any tip.
Well, thank you so much. That was a really interesting talk. We are out of time for questions at the moment. If we could give another hand. Awesome. And our next speaker is James Mickley. Right here. And welcome him to the podium, please.
Okay, thank you. I'm James Mickley. Good morning. And I'm going to be talking about a tool called Vouching. And this is not my work. I didn't write this. This is written by my co-author, Will Weaver. And so all credit should go to him. And so I'm going to talk to you a little bit about this from the perspective of a user coming from Oregon State University Herbarium, where we're starting to use this now. So Vouching is really three sort of components. And so the first one here is Will Weaver has a tool called Leaf Machine 2 that he wrote that finds the labels in an image. And it makes a little label collage like what you can see on the slide here with all that textual information. And so we send that label collage to OCR, and there's a variety of different options for that OCR. So both paid sort of APIs like Google Vision, which is mostly what I've been using, and free things like Florence 2. And so you can stack these things, use multiple different OCRs at once. And so that gives you the text that's in that collage of labels. But that's not really that useful on its own. And so the game changer for me is taking that text and giving it to a large language model, an LLM, and saying, okay, can we categorize this text into Darwin core fields to actually get that data in usable format? And so Voucher Vision supports a bunch of different models, Google Gemini, ChatGPT, other things like that. And in addition to categorizing this data, those models can do some other cool things. They can do unit conversions, they can convert between formats, they can even potentially infer some missing data. So for example, if we have a label that doesn't have country on it, the models can sort of figure that out based on the locality. And so in short, it takes a label like this and it creates structured text, structured data fields like that. And so the large language model looks at this text and it says, okay, this is the collector, and that's the collection date, and that's the genus, and so on. And it puts them into the right fields. And it does this all in a few seconds for a pittance of a cost. To me, it's almost like magic. Okay, so we started using this tool as an experiment at our herbarium, and our first sort of trial of this is to just throw a whole bunch of new accessions that we just brought into the herbarium at it. And so these were mostly things that were collected in the last decade. And so the labels were fairly uncomplicated, but it's a pretty good kind of snapshot of what we're doing a lot of for transcription. And so after running this through Voucher Vision, I did a little bit of data cleaning in Open Refine. And then we ran this through human quality control. And so Alex, my co-author, went through each of these records and fixed any errors and added additional data that we didn't ask for. And so then we compared that curated quality control data back to what we originally got from Voucher Vision to ask how successful was this right out of the box. And so that worked pretty well. And so next, I wanted to know, well, which large language model should we be using and how should we be using it? And so I built this sort of hand-curated benchmark data set of 50 specimens that was kind of designed to be diverse and difficult. I wanted to stress these models. And so these specimens include specimens with a bunch of different annotation labels on, handwritten labels, things like hybrids, complicated taxonomic names, complicated coordinates like taxon range and section and UTM, international specimens that had non-English on the label, anything I could throw at it, basically, I tried to put into this. And so then I ran this through 6 different large language models and compared the results. And just a bit about this data cleaning that we did in Open Refine. It was really basic. I'm sure there's more opportunity to do more of this, but first thing we did was we transformed data in all caps because some labels have that data and we don't want it in all caps. Next, the models are a little inconsistent in terms of how they represent no data. So it could be a blank field or it could be the word none or something like that. And so we did some standardizing of that. We standardized some locality names, particularly country. So labels have diverse ways of writing the same country. And so we tried to make them all the same and match them up with our database. And then finally, we had a lot of extraneous data from some models in the infra-specific epithet field that we asked for. And so it specifically tended to put taxonomic authors in there. And so we wrote a regular expression to try and clean that up. And that was about it. And so here I'm going to show you our results. And so this graph is showing the percentage of fields or percentage of records for each field that did not get changed during quality control. And so really long bars are good. It means we didn't have to do much work. And so for a lot of these fields, the models did pretty well on their own. This is straight out of Voucher Vision before we did cleaning. And so down here in red, the total bar is kind of the combination of all these fields. And so we hit an accuracy level of just over 80%. And so this percent unchanged is kind of a conservative estimate. Will Weaver's messing around with Levenstein distance that kind of shows the amount of change. And I think that's going to be really informative, but I'm just really interested in this from an herbarium curator standpoint. I want to know how many fields do we have to fix. Doesn't matter if they're even small fixes. So that's before data cleaning. This is the same data after running it through OpenRefine. And so a number of fields get longer bars from this. A few really stand out. So infra-specific epithet really improved here from doing our cleaning and cleaning out those taxonomic authors. And this associated collectors field is almost wholly based on standardizing indicators of no data. And so that really helped there. And that brought our accuracy close to 90%, which for me is fantastic. And so this is saving us a lot of time to the point where I think we're going to move forward using this as our transcription workflow. And so previously we had trained, experienced students spending about 5 minutes a specimen. And the key word here is trained. So it's hard to get trained, experienced students and even harder to keep them for any length of time. They're students after all. And so now we're down to about 2 minutes per specimen instead of 5. And so we've more than halved our cost with this. And most of that is still quality control, which we're hoping as this improves, we can reduce further. And so the actual cost of using voucher vision, this little slice in red, is just a few cents. It's really amazing. And so we process about 3,000 specimens a year. So real world implications of this is saving us about 150 student hours and $2,000. And there's a side benefit in that because voucher vision is fairly quick, we've got some data of dubious quality available and searchable immediately, whereas before we would have to wait months to get that stuff accessible. So that's really great for us. Okay, so now I'm going to show you comparisons of these different large language models. This is with the tougher data set. And so the percent unchanged fields goes down a bit. And so we used 6 different models. There's a lot more that are supported. These are towards the lower end in terms of cost. And they're from 3 different companies. So the blue here is Google models. The green is OpenAI, which makes ChatGPT. And the orange is Mistral. And what I want you to really take home here is that there's not a whole lot of difference between models. They've all got accuracy levels between about 68 and 78%. The Google Gemini models up here went out, but only just barely. And a side note, our first test of this was using this Text Bison Palm Google model that was state of the art a year ago. So these things have really changed quickly. So now I'm going to break this out by individual fields. And I wanted to know, are some models better for certain fields than other? And the answer is yes, to an extent, but there's not a lot of difference here either. And so model choice isn't super important, I think. And I think you can make decisions just based on cost and convenience here. But I can highlight a few of where there are differences just to talk about what these models are doing. So first I'll highlight country. And so our Gemini 1.5 Pro does really well here. Some of the other Google models less so. What's happening there is that these are labels that don't have country on them. And the Gemini 1.5 Pro is inferring the country, whereas the others are either leaving it blank or making a mistake and putting a state or province in there. Associated collectors is kind of an interesting issue in that here it's queuing in to other person names in the label text, particularly from annotation labels. And so annotation labels really confuse the model. As voucher vision is written, there's no spatial awareness of what kind of label this stuff came from. And so it just thinks that those are associated collectors. And event date is kind of a similar problem. It's taking dates from annotation labels, or it's having trouble formatting the date into the format that we asked, year, month, day. Okay, so just very briefly, some lessons learned. We've learned that some data fields are trickier than others. Taxonomic data fields in particular are kind of complicated because of all those annotations, for example, but also hybrids and infra-taxa can confuse the models. and it's hard for models to figure out what's part of the taxonomic name and what's an author. And we found that splitting names into component fields seems to work better. So if we just ask it for genus and just ask it for specific epithet rather than the whole name at once. There are some fields that VoucherVision initially supported that we just decided weren't worth asking. So VoucherVision said, if there are coordinates on the label, convert them to decimal latitude and longitude. Sounds great. And so that worked some of the time. And sometimes the model would hallucinate coordinates that didn't exist. So it was essentially georeferencing the model for, or the specimen for us. Sometimes it was right, but also sometimes it was wrong. So that was a problem. We had similar issues with elevation. So we just didn't ask, and we have our students do that. Another lesson, I think, is that the model instructions or the prompting, which is editable, can really be improved, and nobody's really done a whole lot here. Field names and field order, or the order in which we ask for a field, can matter for some large language models. As an example, we asked large language models for associated taxa, which is the Darwin core field name, and that didn't work as well as associated species. So sort of self-descriptive field names really help the model kind of cue in on things. And then lastly, we found that workers are much more used to data entry than data quality control. And so it's easier to fill in an empty field than it is to notice a mistake. And so that's something that we're still working on. So with that, I'd like to acknowledge Alex Kowalski, who is my co-author here, who did a lot of the quality control work and also kind of cueing in on these systematic errors. And a big thanks to Will Weaver, who has created this software. And so he's going to be giving a recorded talk in the Demo Camp 2 session tomorrow. And so please go to that, and you can take a picture of this slide and go to his GitHub. And so find me afterwards, or if I have time, I can take a question or two.
Hi, we have time for one question.
Woohoo.
Go for it. I will allow it.
The first one's an easy one, hopefully. So the first one was just out of interest, why is locality doing so, not badly, but less well? And the other question was, what about reproducibility? How's that looking? Did you do any tests on that as well?
Okay, so two good questions. Locality is lower, and the more textual fields are lower because there's more things that can go wrong. And so even if they're off by one word or slightly wrong, in the way that I'm doing this, they'll get knocked down. So it doesn't mean that they're completely wrong. So that's good. And the second question was reproducibility. So I don't know. My sense is it's not too bad, but that's probably going to depend on the model.
Awesome. Thank you. We don't have time for any more. If there are other questions, I will download the chat and put them in Oxford Abstract. And I believe you can get people's emails if you have any additional questions. And just one more comment, would really suggest watching Will's video. He is an absolute pleasure and really knows what he's talking about with VoucherVision. Okay. So our next speaker, is Fabricio Rios-Montero, who will be talking about the relation extraction from unstructured species descriptions using TaxonNERD and Llama 7B. I do see you in the Zoom, and I asked a quick question. Are you planning on doing it live or using a recorded video?
Good morning, everyone. My name is Fabricio Rios-Montero, and today I am presenting our project on relation extraction for unstructured species description using TaxonNERD and Llama 7B. I am representing the Instituto Tecnológico de Costa Rica, and this work focuses on how we can use AI tools to better understand species interaction, particularly feeding behaviors. To start, it's important to understand the foundational concept behind this research. Data ontologies are crucial because they help structure and link biodiversity data across different studies by mapping information to meaningful concepts. Data alone has no inherent meaning. is the ontology that provides the map linking data to well-defined concepts, enable better integration, application development, and collaboration. At the same time, natural language processing, or NLP, allows computer to process human language, which is critical for extracting useful insights from large amounts of text. In this project, we focus on two key NLP techniques that form the backbone of our research. Name entity recognition plays a critical role in identifying and categorizing important entities within text, such as species names, location, and other relevant biodiversity terms. By pinpointing these entities, we can organize unstructured text into more manageable components. Relation extraction then builds on this by identifying the connection and relationship between those entities, such as feeding interaction or habitat association. This process involves structuring the relationships into meaningful data that can be analyzed, modeled, and visualized for deeper insights. These methods are crucial for transforming the vast and complex unstructured data into a structured format that is easier to interpret and use for biodiversity research and conservation efforts. The tools we use in this research include TaxonNERD and Llama 2 7B. TaxonNERD is specifically designed for recognizing species name and taxonomic entities in biodiversity text, making it highly effective for biodiversity research. Llama 2 7B is a large language model for Meta AI. Help us extract complex relationships between the entities identified by TaxonNERD, such as feeding interaction. Our dataset is sourced from the National Biodiversity Institute database, which contains around 32,000 species descriptions. For this project, we focus on a subset of 200 animal species. The Envio dataset includes fields such as species scientific names and various types of description like feeding, habitat, reproduction, behavior, and more fields. The descriptions are in both Spanish and English, adding diversity and complexity. However, the text often includes related details requiring careful filtering. We select feeding relationship for this project to ensure clear and consistent evaluation. Here's an overview of our process. We begin by collecting and processing a species description from Envio, which includes thousands of records detailing various aspects of a species biology and behavior. These descriptions are then fed in TaxonNERD, a specialized tool designed to recognize relevant entities such as a species name, feeding habits, and other important biodiversity terms. Once these entities are identified, we apply Llama 2 7B for relation extraction. This model is particularly focused on detecting feeding relationships and interaction between the identified entities such as predator-prey, dynamics, or dietary preference. After obtaining the initial results from Llama 2, we realized the need for a simpler and more efficient method to verify and refine these results. Existing software for text annotation was either too complex or lacked the simplicity we needed for capturing the start and end position of entities and their relationships within the text. To address this, we developed our own text annotation software tailored specifically for our project needs. This tool allows us to precisely mark the beginning and end characters of entities and relationships, ensuring that we could accurately evaluate whether Llama 27B was correctly identifying these relationships. By incorporating this custom annotation tool, we were able to refine the model through additional training and evaluation cycles, further adjusting parameters to improve its precision and reliability. We have tested the model with a sample of 200 species as shown here. The overall bird scores are precision at 0.68, recall at 0.73, and F1 score at 0.71. The scores reflect good performance, particularly in recall. The distribution graphs show that most of our scores are between 0.6 and 0.9, but there are some outliers, especially in precision, where the model could be improved. Moving forward, our focus will be on refining the model to address these outliers and further enhance accuracy. Here are some examples of extracted relationship. For instance, from a description about Choroniscus goodmani, the model identified that it feeds on nectar, fruits, and insects. Another example involves Sarcoleus montezuma, where we detect its feeding interaction with invertebrates, vertebrates, and various plant species. These results demonstrate the effectiveness of our approach. Looking forward, there are three main steps we plan to take. First, we'll expand our dataset by processing more species description, which will improve the model accuracy. Next, we will fine-tune the Llama 27B model using the initial results to enhance precision in extracting feeding relationship and reduce errors. Finally, we aim to broaden the scope to include relationships beyond feeding, such as habitat interaction and reproductive behaviors, making the model more versatile for biodiversity research. I want to sincerely thank all of you for your time and your attention today. It's been a pleasure sharing this research with you, and I hope it has provided some valuable insights into how we can leverage AI to better understand biodiversity. I'm happy to take any questions you may have, and please feel free to reach out if you would like to discuss further. My contact information is on the slide. Thank you once again.
Awesome. Thank you so much. Can you hear us on the Zoom? Like you can unmute your mic to see if we can hear you. Mhm. I'm going to see if he's able to take questions. Sorry, give me 1 second. I'm not sure that the speaker is able to take questions right now. So if you do have questions about this presentation, I would suggest reaching out directly. Like I said, any questions that you do have, I'll put into Oxford Abstracts. But yeah, so let's wait just a minute or two before moving on to the next presentation, unless I hear something. if you want to share some of those. We do have a couple of interesting comments in the chat that we are going to share before our next talk.
So the chat for this particular session is running and is quite interesting, and it will be pasted into Oxford abstracts. But just for now, there is a question from Matt Yoder. For those trading against the data, has anyone tried using the Plazi corpus for their XML markup?
Question to the room. Anybody can answer.
And then there's discussions about different training models, and there's a suggestion that the Gobi training corpus could be used.
Okay, sorry, just one more minute and then we'll switch over. Just take a moment of silence and enjoy the peace for a second.
So I can't stand silence. I've played a bit with Plazi. Has nobody tried Plazi at all? Plazi, which is text mining publications, the quality is dubious. The quality for plants is quite shocking, just based on what I've looked at. The quality for animals is variable. So I guess it's one of those things. It's an extraordinary resource, amazing project. I think you'd want to be careful using it as training data. The errors are occasionally fairly shocking. Donat's not in the room, is he? I'm safe? Cool.
Awesome. Okay, cool. So we are going to move on to our next presentation, which is Sanson Poon. And he will be talking about automatic detection and identification of calcareous nanofossils in chalk using deep learning, a proof of concept study. So welcome him to the podium.
Yeah, thank you so much. So after listening to like a few LLM talk, this talk is nothing about large language model. It's more about computer vision and how we use deep learning to enable like more like research using our museum collection. So here, like this talk is going to be about like how are we going to study like chalk and the thing that is hidden in chalk sample. So the Natural History Museum, like where I work, we have a huge collection of like chalk sample, a lot of them, and we have collected it since like Victorian time. And in case you don't know much about British history, Victorian time is around 150 years ago. So it's a very historical collection, and we have a huge amount of them that is sitting in the center of London. So one very important thing is that like researchers want to know from this kind of chalk sample is like when this chalk was formed. However, as I mentioned like 30 seconds ago, this kind of like collection, this kind of like data is collected like 150 years ago. And at that time, people don't really care much about the details of the sample itself. So for example, this is a very typical example that we have in our chalk collection in the Natural History Museum. So you see there's a lot of chalk, very nice. The only information that we know that is chalk that is from England, which like, well, thank you, but like not very useful for like researchers nowadays, because like we want to know, okay, when they were formed, which stage, which zone, which subzone it was formed. But like just giving us chalk of England tell us pretty much nothing. So how can we do it? Instead of like sending it to a laboratory, spending like millions of pounds to try to find like the carbon-14 or all those like radiation way to know like when it was formed, is there an easier way, or is there a more easy way that you can just do it maybe even in-house in the museum? So again, as I say, chalk of England didn't tell us any information. But when we look into the chalk itself, there's a lot of like very, very tiny thing, which is what we call nanofossil, that is embedded in this chalk sample. And by looking into like what kind of like species of like nanofossil that we have in the chalk. The combination of like this kind of nanofossil can actually tell us a lot of information. And one of the most important information that we can know is when it was formed, like the stage, the subzone, all these kind of things. So looking into it gives us some information. How are we going to look into it is, well, from the collection, we take a very small sample, less than a gram, so we're not really destroying the specimens itself. And then we can look in like under a microscope, and then we can find like an expert trying to know, okay, it contains species A, contains species B. So we can map it to like this timeline, and then we will have the result. Easy, right? Things are perfect up to this point. However, this is the challenge. This is how it looks under the microscope. Just a quick question for everyone here. How many nanofossils is in this image here? Who say there is like less than 5? No one? Who say there's more than 100? Okay, sorry, I set your trap for you, but like it's actually around 15 to 20 of them. But it's still, you can see how different they are because they're different species. And it's just, they are just everywhere. They all look different. And a normal person like me, I'm a data scientist, I have no idea, like, how can I find them in a slide? So you need an expert sitting in front of the microscope for the whole day to analyze maybe 10 of this image. Very slow process. But we developed a way to label the data. So within a year, we can manage to label more than like 100,000 of them. That is an in-house labeling tool that we developed in the Natural History Museum. So this enables us to have or try to build an AI-ready dataset that is up to the standard that can be used by AI training. So with this dataset, we will be able to like use computer to look at the image and then classify them, like what kind of species is in there. And once we know what species is in there, with all this combination mapping, directly we will immediately know what stage, what zone this kind of chalk is formed. So this is like one of the very first use case of this data set that we're trying to build, that we're trying to build a data set that is post-Victorian time. So it's like more care about like the future. So with the data set and the AI model that we train and we develop, our data, like our model can actually do quite well compared to like other model that is out there, exists in the market. And we definitely outperform like a lot of them. And at the same time, the number that I report here, like from our model, is actually tested on unseen data. So we don't have to worry about overtraining the model in this stage. So our dataset enable like the use of AI by just like labeling them, and using this also enable another chance of doing research. For example, like here's one of the work that my master student is working on, Evelina, very smart student. We use the data sets that we collect to try to study what is the climate change related to the nanofossil itself. So there is always a big debate in the paleontology community is like, do nanofossil change like with respect to the climate? So there is a big debate say that, okay, things might not change, we just stay as it is. Or like it will change the size, the shape throughout like history because like change of, let's say, temperature, the CO2, like abundance, like, so many different factors, we don't know. So there's different hypotheses that came out in the research area, but like, no one can go into do any statistical study because, well, first, no one has the data set, and second, even though you have the data set, you need a person sit in front of a microscope for hundreds of years to have enough data to do any statistical comparison. But with our method of like labeling the data and with our method of like building a data set that is ready for AI, this becomes possible. So using AI, we can easily segment like what nanofossil is in the slice image. And then once we know that, we can have the bounding box, we can have like the boundary of the nanofossil that is predicted by the deep learning model. And with that, we can immediately know what is, let's say, the major acid, the minor acid, the area, and the ellipticity of the nanofossil. So very easy. We can just put it to the computer, let the computer do the work, and then we will have a distribution of all these parameters. And comparing these parameters, knowing like this nanofossil or like this chalk sample is coming from this specific stage, we will be able to know, okay, we will be able to get distribution, let's say, for Cenomanian stage versus like anything that is after Cenomanian stage. And Avelina's work like suggested that like there is some statistical difference according to the KS statistical hypothesis testing, that like the minor acid and the area have significant difference between like these two different stages. But at the same time, there's also some other like parameters, for example, the major acid and how ellipse the nanofossil is, like don't really have a significance in the results. But what is more important here is like it's show that with an AI-ready data set, it's not only fit for one use case. With like this kind of like AI standard data set, it actually opened the door for a lot of other research and answer a lot of questions that it was hanged in the research community for many, many years and make things possible. So what I've presented here is not much about magic. No magic is involved. It's just like we create a data set with like, it's an AI standard data set, and then we apply computer vision, deep learning. We already know immediately what stage the chalk is coming from. And at the same time, it's also enable other use case of research. For example, answering like, does like any nanofossil have like interaction with like the climate change? So all of these kind of things is just happen because we think a little bit further, a step further when we build a data set. So when we collect data or when we digitize data in any of our collection, maybe it is worth thinking a step more, what is the next generation need? Because I don't want people, let's say 100 years later in Tech Week in Okinawa again maybe, and then people, there's someone like me standing here and say, those people 100 years ago, they just have a data set that just tell us, okay, chalk of England. Well, that is not what a researcher should do. For us doing research, we want things to be improved. So yeah, I think this is just example of like the museum work that when we collect data sets, we think a step further. Thank you.
Awesome. So we have 2 minutes for questions, and come on down.
Yeah, super great talk. Sorry, I think I missed an important point. How did you compile the original data set? Do you use simulated data? And how many classes of these nanofossils do you have? How many do you use all in all? So I wonder how you get the data together. Do you start with human annotation and use then simulated data or something like that?
Yes, so we have one expert in the museum that do all this labeling. So all the labeling like that I show, like more than 100,000, now it's actually over like 300,000 labeled image, is only done by one person. So it is 40 times quicker than any other data set that it was built before our pipeline, before we use our pipeline. So yeah, so there is 60 different species that we label so far, but at the same time, there's around 200 species of nanofossil that is going to be kind of useful for like not only defining like what stage it was formed, but also like other type of research as well. So it's an expanding data set and it's still going on. And one more thing that I can add on top of this is like now for the commonly seen nanofossil, we don't need the human expert to label them anymore because the AI model will be able to generate new label for us to extend the data set. Thank you.
So any other questions? Okay, awesome. Thank you so much for your talk, and let's give another big hand. Awesome. So our next speaker is Nian, and she will be speaking on AI species identification using image and sound recognition for citizen science collection management and biomonitoring, from training pipeline to large-scale models. So thank you.
Thank you. Yeah, thanks for the introduction. My name is Nian. I'm from Naturalis Biodiversity Center in the Netherlands. I'm myself not an AI expert, nor a biologist. I'm here to present our AI species identification work on behalf of our team, which is listed here. So why do we use AI for species identification? Nowadays, biodiversity data are being generated at unprecedented rates from wildlife cameras, citizen science observations, digitized collection data, and research-generated data. And AI can help us make sense of huge amount of data in an efficient way and automate tasks. At Naturalis, we do develop many different kinds of AI models for species identification for different type of applications. And our largest model is for citizen science data, which we call it the multi-source model. I will explain further in this presentation. This is also the focus of this presentation. But we also have developed many other species identification models for example, for insect camera for automatically biomonitoring of insect population. We have several collection data, collection models for museum collection identification, and we are also started working on sound recognition models with sound recognition for birds, European birds, and some other species groups to come in the coming years. Okay, so why do we want to develop a so-called multi-source model, which is short for MSM? We started our first projects in applying AI in biodiversity back in 2017. So initially, we developed many small AI models based on one single data set and for specific applications ranging from citizen science, biomonitoring to collections. Our most successful application has been citizen science, and this is where we get the most data to develop better models. So back in 2022, we had at time developed the two separate models for two biodiversity portals in Europe, and these two models are also used indirectly by several other biodiversity portals in Europe to make AI species identifications. This presents some challenges to scale because we have to train multiple models based on multiple data sets, and we have to maintain this model for different versions, have separate APIs, and this is a challenging in terms of scaling for resources and capability, and also it's not very efficient. So, and plus the fact that we have limited data set for each of the model. So back in 2023, last year, we decided to make a pilot to combine all the data sources from 6 different European biodiversity portals, and so develop one large model for all these portals, while at the same time still offer specialized customization for each of the biodiversity portals. So this has developed into auto-scaling web service based on the API. In the last 12 months, so August 2023 to now, we made 65 million identifications, and it's growing. It's right now being used by several more biodiversity portals in Europe, and also directly linked to the apps they developed themselves. So here shows the training pipeline for the multi-source model. The first step is to collect the training set data from each of the biodiversity portals. And that's amounts to 45 million images of more than 100,000 local taxon names, because each organization has their own taxon names for their data. And we used a customized software called Taxon Map in order to map all these diverse source of data into one shared database. And this contains 35 million images of 41,000 unique taxa. So using this shared data set, we trained a deep model. This consists of one main model and 8 species group submodels. So in total 9 models, and this we call it the shared model because it's ready to use for all the biodiversity portal that provided data, and it's being trained on the shared data of all these portals. However, for some of the biodiversity portals who have for themselves provide large amount of data, we also provided a customized model for to adjust by doing fine-tuning of the shared model to adjust, make a customized model for their own species distribution and their own taxonomy. So the result of this model is a large-scale hierarchical model that consists in total 39 specialized models. So 4 times the customization of the shared model, plus for the insect species group, butterflies, insects, and true flies, we also have a life stage model because these insects can look very different in different life stages. The MSM model developed in 2023 showed a significant performance improvement over the previous model that we developed using the data set of only one biodiversity portal. So here shows the test results of the improvement of the 2023 model to the 2021 model developed only with the data from observation.org. And you can see that not only we have the new model can identify a higher number of taxa, but also across all the species groups, the performance of accuracy has been improved. And also, you can see the average recall, which is an indication of how well the model can identify rare taxa, has been improved. This thanks to the fact that we can pull the data from different biodiversity sources. So if for specific rare taxa, if for one organization they only have a limited number of images, while we can have some other images from a different organization, we can pull the resources to improve the performance. In this graph, we show the data imbalance of, in example, for the Norwegian spiders. On the left side, you have the more common species, and on the right side, the rare species. And the gray area covers about 95% of all the observations. And you can see that when the species is for the taxa that are rare, the performance decreases because we have much less training data for the model. However, the new model still showed 5% improvement of the accuracy across all taxa. And for the rare taxa, 11% higher improvements for the rare taxa. Continuously, we are also trying to develop some advanced feature for the species identification. In 2023, we have implemented the auto validation. This is by means of careful calibration, we can provide automatic validation of the part of the data with low risk. This reduced the need for taxonomic experts to review the AI identification afterwards. This year, we are working on trying to provide taxonomic outputs and the prediction probabilities at all different taxonomic levels. Currently, the model would provide you the probability prediction on the most specific level, which is usually the species level, but we would like to provide it on all taxonomic levels. Another thing we are working on right now is to provide more life stage prediction for the other species groups. And next year, we are going to implement context aware. This is to use metadata, such as location time and the neighboring species to improve the prediction and to reject invalid input, and also to provide image search for similar images. So this training pipeline we have implemented for citizen science data because this is the largest amount of data set we can get, but it can potentially be also used to develop large-scale multi-source data for biomonitoring, also for museum collection identification and sound recognition. So thank you for your attention. I'm happy to answer some questions, and here is our team. And if there's any questions.
Awesome. If there are any questions, please feel free to come up to the mic and ask any. Do we have any online, Robert? Okay, cool.
So there's a question online from Alan Stenhouse. The question is, how does this compare to BioClip?
I'm actually not aware of that one. Sorry.
Maybe one just for my interest. You mentioned the life stages. Are you doing this for plants only, for scans, or also for other species?
We did that already for the insects. So from like egg, larva, and adults, we are planning to do that also for other species groups, including plants. Yeah.
And I guess like this is kind of a follow-up question to that. So for the life cycle of the plants, are you just looking at age? Or do you know if phenology will be incorporated to the plants?
This is all depends on the availability of the data. So we basically collect data from our data suppliers. And if their data includes all this information, then we can implement that.
Awesome. Thank you.
Yeah. Yes.
Thank you very much for your talk. One question, we use a lot of observation.org in our museum, and it's a Münster Natural History Museum in Germany. And my question is, you have shown that you plan to discard some of the invalid data, and you plan also to discard some data on museum specimens, as far as I understand.
Yes, so that's currently not implemented, but it's because sometimes you get people taking selfies trying to identify themselves as
yeah. Self is not a problem. The problem is I was planned to give a talk on Thursday for using observation.org for digitizing, or at least partly digitizing, museum collections. So if you plan to discard this next year, then it's a bit of a problem for us. But yeah, that's why I wanted to ask.
Okay. Yeah, I know that it actually works on collection data reasonably well, but maybe that's something to, because this is a feature we are going to offer, and we only work with organizations. And if observation.org can have an option to switch it on and off this specific feature, maybe that's a possibility.
That would be nice. Oh, thank you.
Yes, thank you.
Thank you very much for your talk. And I asked about train the data. So do you have a plan to add the AI identified data for the train data?
No, so we only use experts validated field observation image for training. We don't use any AI generated data for training.
Okay, thank you very much.
Yeah.
Awesome. Do we have another question? Or, okay, no. Okay, cool. Let's give another big hand. So once again, we do have 15 minutes of discussion. If anybody would like to bring something up or discuss a point related to AI, unrelated to the talks, please feel free to do that. But yeah. Any things people want to say? Oh, yes, go for it.
Hi, I love these talks. I am from South America. And I wish so much we could have similar initiatives with biodiversity in South America. So I'm wondering how we could narrow this gap, because it's wonderful you could have you could identify plants, animals, etc., from the global north, where they've been doing research for hundreds of years. So that's my observation.
Without giving too much away, I know, like specifically in Tadwig, there is a recognition that we need to have better partnerships with the Global South and South America, and there are some collaborations that will be announced on Friday that I think would be really helpful to going forward with like building these bridges between communities, between countries, and getting equitable data, because a lot of stuff is global north centric. So yeah, stay tuned unless other people have things to say.
There's a question for James about your transcribing work. I'm assuming that you're paying your students and therefore the cost outweighs. So if we're doing sort of a similar project where we're using AI to capture OCR, and then we're using AI to kind of turn things into data sets or into spreadsheets that we can then give to volunteers to check. And I've been getting comments back from the volunteers, oh, it's making stuff up, there are too many errors. You made the comment that it's quicker and easier sometimes to enter the data from scratch than it is to use AI. I'm just wondering, if you're not paying your students or the people who are doing this work, do you still think it's worth using AI to do this kind of initial data collection and formatting?
Yeah, good question. So we do use some volunteers. They tend to be even more transient and less trained than the students. And so that ends up creating more burden for me. They also don't love transcribing. And so they're more likely to want to do specimen mounting or something more interactive. There's some exceptions to that, but that's been an issue, I think. And we've used Notes from Nature, which is community science transcribing. And that works to a point. There's actually 3 people that transcribe every record, but they don't follow directions. And they're also not trained. And so that, it ends up being more overhead for me.
Okay. Yeah, my experience is different. I seem to have the best volunteers in the world. Many of them have been working on our project since it began in 2010. This is BHL Australia volunteers, but also we've had incredible experiences with Digivol community, where I would upload like a diary on the Friday night and it would be done by Monday morning, and they would have asked all sorts of really good intuitive questions, and I would answer them and they would clean up their own work and we would get an outstanding result. And I have a team of volunteers who began doing a whole lot of data generation for us during COVID. They're still doing that work. I can't keep up with them. So we looked into using AI and we were trying to use a paid service. And again, it was like $13,000 to do what my volunteers would do for free in a couple of weeks. So, but yeah, they've come back to me with these, there are too many errors and it's making stuff up. And could we just do it from scratch? So I'm grappling with that right now.
Yeah, possibly. But I just did 5,000 specimens for $5.
Yeah.
So, I think the approach that we're using, where we're using quality control, but sort of doing half of it,
yeah,
if you can train people, is still worth looking into.
Okay.
Then we'll go to you, and then we'll go
I wondered, just on the topic of, you said that maybe people don't like doing data correction as much as like de novo data entry. Do you think that's a human-computer interaction problem? Like, we haven't got the right interface that makes that a compelling task for them to do? And have you got any thoughts about how we could improve that?
Well, I would say just data entry and data correction at the same point, but I do think there's a human-computer computer interface issue here with, doing better corrections. And Will Weaver's actually working on some of this, basically forcing people to confirm things through an interface.
You said your students were seeing things being made up, and I was just wondering, what if you didn't take the LLM approach and took the other approach of more OCR things, like James talked about right in the first slide, like Google Vision and how it was working with that.
I think that's kind of the approach we are taking. So, I mean, certainly the AI tools, like we're scraping a lot of data, which gives us author names, and the author names are not in the format that we want. Certainly my experience with ChatGPT and saying, can you take all it, like the author names are in variable formats. Some have initials, some have names, like some are Spanish names where I actually don't know which is the middle name. And like ChatGPT has been incredible for that. Like, can you take this list of authors in all sorts of different formats with all sorts of different types of names and format it into exactly the format we need? That is a time saver that the volunteers are so grateful for. And certainly as well, there are other tools we're using AI for, but it is making up stuff. So yes, we're doing the combination, and yeah, I'm still so grateful for the AI time-saving mechanisms that we're using, and so are the volunteers. But yeah, they've just got back to me today going, yep, another lot of there's a whole lot of made-up things in here, and also gaps that it should have known about. And so I think again, I think I'm being trained by ChatGPT as well to ask better questions and to actually say, don't add anything, do not make anything up if it's not there in the text. And that's getting better and better. So the volunteers and I are learning from each other and learning from the tools.
So my question actually follows on really well from that. And that is it a way, or are people already doing this, where we're sharing and collaborating on improving the prompts? Because that's going to be such a key thing. I mean, I've been doing some testing myself and it makes a big difference. But I think that would be really nice if we could find some kind of mechanism to share and collaborate on prompt writing.
I know in general, there's a lot on prompt engineering. I don't know of a specific repository for biodiversity specifically, but in general, the computer science community and the LLM community has a lot on prompt engineering. Also, Siobhan, do you have something to say?
So this question was in relation to your OpenRefine work. I'm assuming when you put the collectors' names in the collector field that they're name strings. They're not identifiers or linked to an identifier or anything like that. Is that correct? Have you considered reconciling your OpenRefine collector names to Wikidata and seeing, I mean, it wouldn't necessarily work for most of it, but you would at least get some ones, especially if you've got quite unique names. Have you considered that?
Yeah, so we're using Symbiota, and Symbiota doesn't track this stuff yet, but they're working on it. And so I'm really, really waiting for them to get that agents module working so that we can do that kind of stuff.
Yeah, well, I've spent my life putting stuff into
Yes,
Wikidata, and I want people to use it. So please reconcile it in OpenRefine, use my work. There's, I don't want to be doing and shoving it all into Wikidata and people not using it. Right. And OpenRefine is built for it. So yeah, I'm begging you to use it.
So we're behind where everything's verbatim off the label.
Yeah.
For the most part.
Yeah,
and it would be great to consolidate that to actual people.
Yeah, cool.
We have a few things on the chat too, so.
Yes. For those on the AI side, what kind of gold standard data sets are you looking for? What do you wish you had for training?
Anything goes.
The text stuff. And there is also discussion on the chat about various other people who are present but virtually talking about they can provide better links to better API services, and the suggestion that we should maybe set up a repo for exactly what Elspeth said for the prompts.
I guess a follow-up question to what Alan said is there a gold standard for AI, generally, data set? I'm not sure because my approach is for it to be really specific, and some data sets that are perfect for one model will just not work well for another model. Is that is there a gold standard, or do we have a lot of gold standards like AI Training Data Olympics. This is the best for this event, and this is the best for that event.
So there is an answer to that online. Gold standards are made for incredible differences. For example, the Netflix one led to major engineering challenges. There can be so many. It just means that significant work went into them.
Cool. Are there any other thoughts, reactions, things to say? Oh, go for it.
So I guess I wanted to ask, the people interested in sustainability of AI. We're clearly seeing some amazing talks. The technology is amazing. It's empowering all kinds of stuff. And there are these concerns about sustainability, which raises the issue, I guess, at what level of sustainability would be bad? At what point is somebody going to say, we're not going to do this? And do we have metrics we can use? So I found a website that said 90-odd thousand ChatGPT queries is equivalent to a flight from, I think, New York to San Francisco. And that's what, $20-30 carbon offsets. Is that the kind of thing we're talking about? In which case, this seems eminently sustainable. Or am I looking at this the wrong way?
Linked to sustainability, but not completely answering the question. But for example, if it was a choice between using ChatGPT API to do the data extraction or training your own thing to do it, then the latter will take up it's much less sustainable to do the latter way because it will require a lot of training. So sometimes you are better off using what's already there, using the ChatGPT API. And that's something that people are thinking about. I know in our team, we are certainly thinking about things like that. What can we use that's already there? What things have already been trained? Can we just do some transfer learning with this and stuff like that? So it is on some of our minds, I know for sure. But maybe when that repo is set up, we can talk about that some more. And also, just to add to that, Slack would be useful as well, maybe in addition to the repo, I think.
Okay, two things. Lena Struve from Rutgers University. So first, talking about sustainability and things, I think we also need to think about not just energy, but also what the result of these inquiries are going to be for biodiversity conservation, that we're going to find new species, new places that these species occur in, and that is going to increase our possibility to conserve the biodiversity on the Earth. For example, in, new places where we didn't know that they existed before, or brand new hotspots of biodiversity. Okay, my second thing is that it really surprises me that nobody really talks about iNaturalist here. And especially for those of you that were from Northern Europe, wherever the speaker is, I know. You're developing this brand new thing from Northern Europe, and you're not using the iNaturalist data set that have their whole new thing that they are using for their AI based on research grade observations that are millions and millions of observation. I use it daily. It is around the world. It's a global thing. It's free. It is using observations from citizen scientists, or what we should call community scientists, but also highly trained experts that are going in and are editing and helping out, and there's an ongoing conversation about this species, that species. Bad photos are not included in what is used as training data. There's no AI-derived training data, and we are engaging the whole community around the globe by using iNaturalist. And that is not even included in the training data set for the Northern European project that you're working on, and I don't really understand why. But. Oh, it is. I missed that then, because it wasn't listed at the bottom. It wasn't mentioned, but it is. Okay, well, great, thank you. Please recognize them too for all they do. Thank you.
Yeah, well, two things on the IA sustainability. Actually, it's a bit to see like an extension or new perspective on what we were discussing already before. There was so much EA around. about for each project reinventing the wheel and reinventing again a same system and forgetting what has been done in projects before. And I see with EA only this phenomena still continuing and maybe amplifying. So I think a general message is to a bit see what is already out there and what works, instead of wanting to do each time your own new development just because it's your new project or because you want it to be from your institution or my system. And collaborate more could also be a solution, but that is a discussion that has been going on long before IA and could also relate to the last remark on iNaturalist. It's working so well, it's working worldwide. It works better and better for so-called cryptic species that you can more difficultly identify. So why not bring your contribution to this wonderful system? And why do you have to develop your own local IA for just your species around you? This is something you can indeed reflect. And when working with citizen scientists, we really prefer iNaturalist to a local system, because if they take a picture of an exotic plant in a garden or in a park, they get a meaningful answer, which they don't get with the local system that just gives something completely wrong or no answer.
No, that's like a super good point. I think like the software community has also really struggled with not reusing like perfectly good software and like, I'm gonna build this Python package from scratch so I can say that I did it. So yeah, it's definitely been a struggle.
Yeah, I'd certainly support reusing software as much as possible. It takes a lot of work. I had a project this summer where we used voucher vision and we proposed some changes to it, and it takes a lot of work to set up a project so that you can accept those changes. And I really don't want to overload Will. He's doing really great work. He's also got his PhD to do and his research career to do. So I think we need to think about the way that we look after people that do those kind of projects and encourage them to develop them in such a way that they are pluggable and modular, so that we can use them as much as possible, rather than we start again and write a new one.
I just want to strongly agree with that. You know, Will's a grad student. This is one chapter of his thesis, and he's doing a huge service for us. He could use some help, and I don't know how best that integrates with his vision, but I'd love to see more people working with that.
Any other final comments or questions? We have like a minute or two. Oh, yep, come
on. Just for the voucher vision and for Will again, I want to recognize a great work that he has done. One of the things that we started to do with Will just getting off the ground is the documentation for Leaf Machine 2, especially as things change very fast and they need to get kind of updated with how to use things. And that's one thing that it has a very low technical barrier. So if you're not a coder or you don't want to get into the nitty-gritty of things, but you know how to use the service, then contributions to that are always welcome. And we do have a repository for that. So if you do have any prompts that work well or setup tips and stuff like that, feel free to contact me or Will, and we can get started on that and help everybody else use the system better.
Well, if there are no other comments or questions, thank you again so much for being so interactive and attentive. We have one more. We've almost made it to the finish line. We have one more session after lunch. And yeah, thank you again.
Yeah, no. It's not in this room. Yeah.
That's great. I actually realized that.
mean, all.
Oh, yeah.
Oh, right. Oh, okay. Ah, cool. Okay. Also, you're joining both. That's right. There can't be many people who join both. You guys love it. Love it.
Hello? Yeah. Hello, everyone. Sorry, we have another session now. We have another session now. LTNG02. Yeah. Sorry. So the session is entitled Diversity and Commonalities of Artificial Intelligence Applications to Biodiversity Issues and Data. I'm Youcef SKLAB, and my Auguste, Gardette are the moderators for this session. Yeah. Sorry. Thank you. So, as we have only, I will do it short. As we have only 13 minutes for 6 presentations, we will start by the presentation first. We have limited each presentation to a maximum of 4 minutes, and we will allocate the 5 last minutes for the question and answer session. So let's quickly move forward for our first presentation entitled The New European Union Artificial Intelligence Act Impact on the Biodiversity Information Community by Patricia Mergen. Patricia, the floor is yours.
So I will briefly present about a new EU regulation that is called the EA Act. So although it's European-based, it has an influence worldwide. As you will see, there are binding rules that it also needs to be implemented in Europe, even if for IA tools that are developed outside Europe. Because it's a regulation, it's legally binding, and it has come in force in July 24, so very recent. But there will be a sort of grace period to be fully implemented in 2030. The main goal is really to protect the humans against manipulation or unwanted usage by EA of their personal data. Its risk is based on a definition of analysis of risk from unacceptable risks, which will be IA tools completely prohibited in Europe, back to minimal risk and no risk and the intermediate categories. Now, there are some general principles that are for all risks level. It's mainly research, and what we do in biodiversity information will probably be low risk to no risk, but with the transparency requirement, our algorithms may be reused for other users that fall in higher categories, but there it's clarified that it will be those third parties who misuse it that will be affected and not us directly. And also there are some uncertainty, for example, border control. They clearly target people identification, but if we have IE tools to identify traffic of illegal species, it's totally unclear if we fall in this high risk or not, because it's leading to border control. And also, as I said, if it's anything developed outside Europe, if you want to use it and deploy it in Europe, you will have to follow the EU rules, and you will have to have a contact institution or person based in Europe. Now, will this be a help of a curse for us? Of course, ethical consideration and manipulation of the information is something we don't want, but as in other cases, will this regulation really help those it should help? We never know. We know that unfortunately it will bring us much more administration, because you will have to justify that you follow the rule, you will have controls and much more templates and things to fill out when you want to get funding. But they can also help to leave some uncertainties. For example, if you want to do text mining on journals, there is now a clause in Europe that they can opt out if they want. And it's totally unclear if we can still do text mining and data mining, even for scientific purposes on journals, where on one hand there is a directive that says we can, but there is also a clause that says that any editor or journal can opt out and say, no, you are not allowed to use our text, even for research. And in the discussion with the EA Act implementation, we're trying to solve this issue and bringing clarity so that we know what we can do and what we cannot do. So I just want to thank all these projects that helped me reflecting on this new AI Act, and I hope we can all together get prepared that we can still do all these fantastic developments, also with our European colleagues, but there will be regulations and rules coming up that I wanted to just make you aware of.
Thank you, Patricia. So the next presentation will be by Nicky Nicolson about deconstructing the monograph, building multi-modal datasets for machine learning. Nicky.
Right, thanks. I'm going to talk about deconstructing a monograph. This is primarily work that was done by my student, Erin Carraby, who worked with me this year on a placement from the University of Bath. It's been a pleasure to work with him this past year and present his work here. So, first a quick tour of what a monograph is. If we talked about them in ML terms, we'd probably call them multi-modal data. So they're the gold standard outputs that we have in taxonomy, and they link these three kinds of data that we've depicted here. So big blocks of text, which describe species in text. They've been written from specimens that we have in our herbaria. And also we get these illustrations. So they've been drawn from a specimen and they depict the characters which are in those text descriptions. And we wanted to see if we could turn these publications into data sets so that people can use them for generative AI. So could they look at the description and the specimen and perhaps make an illustration which shows the characters and states which are present on that specimen, because we know exactly what was used to make each of those outputs at the top that are human made, the text and the illustration part. So what Erin did was we processed a load of monographs from PhytoKeys, and we downloaded all the data, downloaded these big text descriptions, downloaded the illustrations, and he segmented them. So what we see in illustrations is they're very complex figures often. They are not just one illustration in a figure panel, but they've often got like 4, 5, 6 different parts. And they're drawn at very different scales. So we want to break them up into separate images so that we can relate those plus the scale onto the herbarium specimen that was used to make this illustration. So he's animated there the breaking up of the illustration that he's done. So we've got a capsule, we've got a fruiting branch, and we've got a branch with buds, immature flowers on it, and we can pull those out separately, and we know exactly what specimen was used to make this illustration. So we should be able to find these structures on that specimen. The part that I did was the resolution of specimen references. So in the monographs, typically we get specimen references in the form of collector name and number. So we don't get persistent identifiers that we can quickly resolve and get a specimen image back. We get things like NI 1, 2, 3, 4, and we've got to work out where that is. And the method we used for that is I made a Hugging Face space, which exposes a GBIF dataset. and it exposes that, that download as something called a dataset, which is a really cool package, which I recommend you look at or talk to me because I'm here all week. And it stands up a tabular data set, a download, to a place where you can interact with it and you can run APIs against it. So when I stood that up on the Hugging Face space, I made a binomial call, which extracts the first collector name. So then we've got the bits that we need to get the specimen data from. We've got the first collector name and we've got the collection number. And also I made a reconciliation service using a plugin to dataset. So this is all no code stuff, all using APIs from someone else. So now I can fire a collector name and number against this dataset and get specimen references back. This is the kind of thing that Siobhan is gone now, but it's the kind of thing she was talking about in her previous question in the previous session. I had related work from a colleague of mine who's also been working on description atomization using large language models, a bit like some of the talks we've heard earlier before, and we're gonna bring that in. I've got a reference for this work, which was primarily done with the World Flora Online. So there we're taking big species descriptions and breaking apart them into the characters and states that are found in those species descriptions. So we're going to integrate that. You can find all our work on GitHub at that address, and thought I'd list a few reasons why you might want to look it up. You might be interested in specimen reconciliation and standing up this endpoint. We've documented how to do that. You might have ideas for the data set if you're a machine learning researcher and you want to try generative AI on this multimodal kind of data. You might want to use the process on your own, publications or data, or it might be something else. And we've got a discussion board, so yeah, please do talk to us.
Okay. Thank you, Nicky. Let's move forward with Auguste Gardette. We will talk about the project Meta Plant Code, about harmonizing plant meta barcoding pipelines in Europe.
So hello everyone, nice to meet you. I'm Auguste. I'm a PhD student working on the Meta Plant Code project. It's supported by Biodiversity Plus funding. And today I will talk a little bit about this project, and then I will go about the AI component. First, I maybe need to introduce some concepts. So there is this notion of DNA barcoding. So it's like a barcode you have in your shop, but here you're scanning a DNA sequence and you try to identify a species. Theoretically, one barcode equals one species, but we are working with living science. There is always specificities that bring some complexity. After that, you have the idea of meta-barcoding, which is the fact of taking a sample in your environment, your sequence, your region of interest, and then you screen the result against a barcode reference database that is allowing you to identify the species. And what we want to do with Meta Plant Code is to harmonize this whole process for plants in Europe. It is led by Professor Birgit Gemen-Holzer from Kassel University. It's involved 35 people currently in more than 10 countries. And for our part, we are in the charge of the AI things. And so the first things we did is to build like a model able to directly classify the barcode into species. It's supposed to be faster and more robust than classical bioinformatic approach. So the first thing we did is like a kind of benchmark of all of the available model on genomics. What you should notice here is just that they are all performing differently. And if you deep dive, in fact, into the errors, you will notice that they are not making the same type of mistakes. So that means we can adapt to like a ensembling approach, combining all of the prediction of all the models, and we can reach up to 99% of precision. Of course, it has been made on a subsample or off plants. We still need to work for generalization, and it will be lower than 99%. So now we need a level of abstraction. This should be like just a module you have on your the top right of the screen, and it will use only the genomics information. But with that, we will not be able to solve all of the case. And so we want to fill the gap by using the metadata associated with the sample, for example, where it has been made, when, and how. And so we want to use a large language model specialized for understanding biodiversity knowledge, such as species-species interaction, species habitat, and things like that, to embed the metadata associated with the sample. and merge it with genomics information into a multimodal approach. And this should give us another occurrence table. So at the end, we will have 2 occurrence tables from this sample. We expect that the multimodal approach is better than just the genomics one, but we can also compare the prediction and notice if there is maybe it's just error, maybe it's invasive species, or maybe it's new species. And last things, what we are doing in AI for plants barcoding should also be true for other type of barcodes, such as fungi, bacteria, and animals. Thank you for listening.
Okay, thank you, Auguste, for the nice presentation. The next presentation is entitled Threats for Efficient Navigation and Search in Natural History Collections by Marius-Saliba.
Hello everybody, I'm Marius-Saliba of Sorbonne Université and Muséum National d'Histoire Naturelle in Paris, France. I'm going to talk today about traits that can be useful to, traits backed by AI that can be useful to navigate natural history collections. So we now in France recently finished the e-RecolNat project that aimed to get most of the corpora of our national collection available. The issue is that we have a lot of data, but the only way that we have to browse this data is to use the traditional metadata associated with collections, such as species names and our region of collection and stuff like this. Another way of approaching the browsing and collection database would be to enhance these databases with traits associated with specimens or with species that can be found in literature. And this, for example, can be extracted by computer visions for specimens or directly by text mining in the literature. So to make sure that what we will develop in the context of Ecole Plus, the project that took that now is focusing on collections in France, we surveyed the different specialists of different taxa working on with different supports, different type of conservation methods, such as fossils, some specimens in alcohol, 3G type 3G specimens, or 3G extraction of 3G imaging of specimens, such as bones, insects, etc., etc., and obviously plants. Plants have been until now have been the focus of this project until now, but interesting thing were found for all these taxa. And the main result is that the traits that can be used are basically infinite. We can imagine a system that can, for example, let us search in collection based with queries such as, I want all the specimens with red flowers that live in South America with dentate leaves. And this can be linked to morphology, for example, to ecology, everything that can be found directly on the specimens or in literature. It can also be related to human use. So, for example, we can extract from collections all the specimens that are used in pharmacology or ethnobiology, etc., etc. This information can be reached. We have technical approaches that Youcef will talk about. But for the limits, the main limits that we have is obviously one that is very classical in artificial intelligence approaches, that we lack personnel to be able to annotate the specimens that we need, especially that some of this annotation require a high degree of expertise. Even a specialist of a taxon is not always able to go through all the different possibilities or different traits. We have another issue is the one of vocabulary, the one of language. Should we work in French and English? It's a French project, but also even if we work in a given language, how can we make sure that the user can see a difference between, for example, a denticulate leaf, a dentate leaf, a tooth-like leaf, and how to make sure that when he or she is making the query, what we are sending back is what he or she meant when he put in the query. And the last issue that really appeared during the interviews of the specialists was the fact that we are still lacking a lot of metadata, classical metadata in our collection databases, which makes it harder or less pertinent in a way to go through with AI as long as these metadata are still missing. I would like to thank all the specialists that kindly answered our questions. For information, we got around 200 different traits. And I would also like to thank the École Plus team and Youcef and the MISCO team that is going to work on the really artificial intelligence part of the project. Thank you very much.
Thank you. Thank you, Eli, for this detailed exploration. Next, we have Klaus Weyland, we will talk about using WebAFDOs to integrate AI taxon identification and citizen science.
Okay, yeah. Thanks for having me here. Klaus Weyland from Senckenberg. I will tell a little bit about our efforts to combine AI-based taxon identification and citizen science and capture the data in so-called WebAFDOs. The basis on what I will present today is the AI for Wildlife project, which combines AI and citizen science for curation, annotation, and mobilization of biodiversity monitoring data. It leverages on the original wildlife project started by Martin Janssen from Senckenberg, which has the aim to monitor and quantify the faunal dynamics in particular regions, the Chiquantano dry forest in Bolivia, and observe responses of the species there in response to increasing deforestation and land use. This is shown on the right side for two subsequent periods, 2018 and 2022. What you already see there is the increase of the light green areas, which correspond to increasing land use, mostly to use for livestock farming. I must say that all the land you see here in this area is privately owned, so people are entitled to do this. This is not conservational area. Nevertheless, a large community of mammals settles there. 24 mammalian species were identified by the Camera Trap project, among them apex predators like jaguar and at least transiently puma, other carnivores like ocelot, jaguarundi, and smaller ones, as well as several sorts of peccary and tapir. What Martin Janssen does in this project is involve a large citizen science community to analyze the data from the camera traps. He has more than 1,000 active participants, mostly from the Senckenberg Society, who already processed more than 100,000 images, each in 10 to 12 iterations. And what we do, to support this process, is to develop the AI for Wildlife portal, which will be supportive in this project. The key objective of this is to provide an intuitively usable portal shown on the lower left side, and then we add a verified machine interpretable layer to this to enable software agents or in short machines to process the wildlife data in concern. We use a particular implementation path for this called WebAFDO, where we involve several web-based technology, in particular a lightweight container format called RO Crate, to bundle everything about a particular capture event, the original image, as well as licenses attached to the image and details about the sensory data in a web-readable file shown on the lower right side here, serialized as JSON LD. And then we exploit another technology called fair signposting, which associates the RO crate we developed with the image the user can see on the left side. This is, of course, usually invisible for the users. There is a particular tool called Signposting Sniffer, and this is this yellow box, which can be used in Chrome as a plugin to visualize the annotations, and there you can see that you get information about multiple images which belong, of course, to one of the capture events. Usually, if a species is detected, a series or burst of 3 to 5 images is fired up, as well as additional annotations by users and so on. This is all stored in the RO crate and then mobilized via fair signposting with the capture event, with the display of the capture event. Just to illustrate this a little bit more, I'll go with you to a short data flow in the platform. We start with A on the left side. Data comes in as captured image, is then subsequently annotated by a machine learning services, which adds a region of interest in it and does a taxon determination or proposes the taxonomy determination inside. The data can then reviewed in the human usable portal and the human user interface by citizen science. We call this human in the loop. They can revise annotations or redraw the bounding box, improve it, for example. And all these processes, user roles, cryptified, of course, are stored in the RO crate and mobilized for the platform. And data can then, we are not that far, but we will do it, of course, pass back to the machine learning algorithm for retraining to improve the training of that. So this was just to give an idea. Thank you very much.
Thank you very much, Klaus. So now let's welcome Youcef, that is following a little bit the work made by Ellie, and he will talk about a platform able to handle a deep learning analysis for herbarium images.
Okay, thank you. So I'm Youcef SKLAB. I'm from IRD, Institut de Recherche pour le Développement, France. I'm going to present our ongoing work on developing a deep learning powered platform called PlantAI for herbarium image analysis to host the deep learning models that we are developing as part of the ECOL+ project that Eli has introduced. So ECOL+ project is a project which focuses on enhancing tools and methodologies for the study of natural collections. In this project, we are developing a set of models for image segmentation, object detection for non-plant elements detections, and plant organs detection, for example, and also image classification for plant traits recognition and some for management of herbarium collections. We are also working on multi-modal models combining images and text. And recently we started working on developing models for leaf measurements. But we noticed that using these models by experts who are not familiar with AI environments is not easy. So to make it easy, we came up with the idea of developing this platform, a platform which has some features that I will show you in the next coming slides. The first one is we have a model catalog in which we deploy the models that we are developing. And it is easy to deploy a new model as we have just to follow a sample form to specify the type and the path of the file containing the model that we want to deploy. In PlantAI, we can work in a collaborative way. We can create projects, data sets, corpora of images, and share them with other users. So we use also metadata as filters and AI annotations as filters too. In PlantAI, when a model is deployed, experts can use it to annotate specimens in a given project, and then all the experts who are part of the project can review the annotations, leave comments to explain their review if they want. The reviewed annotations can be then used to retrain models later or train new models to improve performances. And once a model is added to the catalog, its annotation capabilities are immediately added as filters in the navigation and the creation dataset interfaces. In PlantAI, we also add dashboards for easy visualization and analysis of specimen data and AI annotations. You can see here an example of the confusion matrix, for example, or the distribution of other annotations, the AI annotations or location here for in a map. At the end, I would like to thank all of the team involved in this development, and thank you.
Thank you very much, Youcef. I think we're supposed to close the session, but maybe if some have some question, feel free to ask.
Youcef, quick question. I mean, do you involve in the annotation for the plants any expert knowledge? I mean, there are floras or other kinds of plant databases, of course, which might already know that a particular species has, for example, serrated leaf and so on. Do you use these sources also in this process, or do you completely rely what you extract on with the machine learning?
For the annotation?
Yeah.
No, we use annotated data to train the models, but after training, we use the models to annotate the specimens. But of course, the expert then can review the annotations.
Yeah, can review, but do you also include electronic sources, for example, digital floras and so on, digitized floras for
during the annotation?
Annotated, yeah.
No, just inference, AI inference. Thank you.
Do you have a way to bring the data back into your collection management system once it's been analyzed, or are you not bringing it in?
After annotation, you mean? Or
Yeah.
Yeah. We do export the data in a CSV file, and then so we can use it to develop other models or to retrain the model and improve performance. It's just to export the data in a CSV file. Thank you.
Well, if there is no question, maybe we can go for lunch. Thank you.